Tracey, can you share screen, please? Actually, I think almost everybody here knows who Tracey is and who actually the other people on this call are. So I'm Peter van Hersden. I'm a senior bioinformatician at the South African National Bioinformatics Institute. Tracey is well introduced here on the slide, PhD student at SANBI, also South African National Bioinformatics Institute, in the lab of Professor Alan Christoffels. And the other committee member here is Martha Luca, who, in fact, I don't know your current affiliation, but Tracey, Martha, and myself have been organizing these events and ably assisted by Olayton, University of Glasgow, of course, yes, sorry, how could I forget that? And Olayton is, what is you are the communications officer for ASBCP or something, right?
Training.
I know you do all of the organizing events and things.
Yes, thank you. It's training officer.
Okay, Tracey, take it away.
Okay, hi. You guys can still hear me, right? Because this microphone is a bit...
Yes. Hearing you
okay,
loud and clear.
I'm going to talk a little bit about the Operon predictor that we designed at SANBI, which we called COSMO. It was initially called Porridge, but yeah, which stands for condition-specific mapping of operons. For the first part, I'm just going to go into the why of COSMO and obviously also of Mycobacterium tuberculosis. And then I'm also going to talk about some of the results that we've seen compared to our comparators. And then in part 2, I'm just going to show a little bit of how we tested the predictor on some other strains from different lineages and under different environmental stress, and then show the results that we've gotten there. So obviously the first question is, why are we looking at MTB? This thing is so old. Initially they thought it was as old as them discovering it in the Egyptian mummies about 2400 BC. But now they think that it actually has been evolving with early humans about 70,000 years ago. And now I'm not just mentioning the age for nothing. I'm going to explain a little bit why I'm bringing in the age of this thing. But during that time, we've had a lot of research that's been into it, and obviously a lot of advancements that have happened in terms of the drugs and the vaccine that has been developed. So much so that this guy, Sir McFarland Burnett, who was a virologist amongst other, he predicted that by the end of World War II, we would have solved all of the major problems that we have with infectious diseases. And this was obviously not an unfounded prediction, because at that stage in the early 1920s, they'd already created the BCG vaccine for TB. And then in the mid 1940s, they also developed streptomycin, which is obviously the antibiotic. So things were kind of looking up. But obviously MTB decided that it wasn't done with us yet, to the point that now currently we're still seeing millions of cases of MTB every year. MTB, Mycobacterium tuberculosis, it's just too long to say. And then we've also been seeing a gross amount of deaths as well, with them predicting, let me just put my laser on quickly, that about 2 billion people are also infected with the disease without knowing it. But I'd like to point out that even with our current research, we've had huge strides in terms of the amount of lives that we've been able to save with the research that has gone into MTB. So the question obviously is, why are we still kind of stuck? Why are we still having this massive problem? And one of the issues is the emergence of drug resistance and the many different actually profiles of drug resistance, because it's not just normal, but also multi-drug resistance up into total drug resistance. And some of the current research that are going into it are mostly, actually most of the research are on a gene-specific level, and some even zoom in too far as SNP level. And we're not here to criticize what's been done. I mean, there are limitations involved, and we've, like I've said, we've at this stage designed so many anti-tubercular drugs just based on that type of research. But there's been some indications that we might have to start to shift our research from looking at it purely on the gene-specific level to the more higher modules, such as operons. One of it is this RPOB gene, which is basically the gene that is targeted by rifampicin, the first-line drug for MTB. Usually, it accumulates sometimes mutations in its gene, and that causes it to resist rifampicin. And usually that comes at a fitness cost to MTB. But what they've observed was, and by fitness cost, I mean that basically affects its virulence. But what they've observed is sometimes there would be mutations in this RPOA gene, and then also in the RPOC gene, which is basically directly adjacent to this gene. And those mutations actually would restore the fitness cost, which we call compensatory mutation. So it then continues to be drug resistant, but it also gets its original virulence back. And then beyond just looking at one or two upstream or downstream genes, they've also seen that in some cases, more than one gene within an operon are mutated, and then that confers drug resistance. And in some cases, all the genes within a particular operon are actually targeted, and mutations then cause drug resistance. So obviously this question is, okay, so hopefully I've convinced you that we should be looking at operons. So what exactly is an operon? I wanted to apologize for showing this animation again, but I like it so much, and I don't think there's anything to apologize for. What an operon is normally in eukaryotes, especially, we have multiple genes in a specific pathway, and they may be scattered throughout the genome. But most of the prokaryotes and archaea, they have evolved this nice molecular tool where they actually place the genes that are needed right next to each other, which essentially allows one transcript. So this is your polytranscript consisting of 3 transcripts to be co-transcribed into a single polytranscript, which obviously allows it to then very rapidly and efficiently respond to the drug stress at hand. And I mentioned age before. There's some, I don't know if you call it speculation theories, that this efficient ability to, on an ad hoc basis, produce operons might be one of the ways in which MTB has been able to survive for thus long. And again, the question then is, how far with our research in operon identification? At this stage, a lot of it is done or has been done with lab work, but that obviously comes with its limitations, and that's beyond just the limitations in terms of cost and labor and its complexity, but also the very nature of operons is that they are not always active. And so obviously we've got all these genomes for which we don't even know much about, and that comes with the problem of needing to design primers. And then it also means that those genes have to actually be active so that we can track them. And so lab work isn't always going to help us in our quest to identify operons. But RNA-seq came along, so RNA sequence data, and this actually allows us to kind of get a snapshot of the entire transcriptome that is available or active at that specific time. It does not need a lot of nucleic acid, in this case RNA, to be present, and it can resolve the nucleotides, or it can resolve reads up to a nucleotide level. So and it's cheaper as well. So a lot of the existing Operon predictors are actually using this RNA-seq data, but even they have a few limitations. So one of it is actually because of the MTB's genome itself. The genomic architecture is quite different. One of the issues is that approximately a third of the MTB genes are leaderless. So, and there's a whole bunch of other things like sigma factors that we don't see in other organisms. So it actually has a very unique way of regulating its genome that's different to other prokaryotes. The other issue is one of the most important features or predictors that are present in other Operon predictors is this, this intergenic region distance, which they limit to because the assumption is that if two genes overlap, or if there's a very short intergenic distance, which is usually below 20 base pairs, then that's a very good indication that those two genes are part of the same operon. So what Remap, which I will talk about in a bit, has found is that this is actually a kind of a horrible predictor for MTB because MTB has many of verified operons that have very large intergenic distances or intergenic regions or lengths between genes. So this is not a good feature to use. The other issue was inefficient parameters that users can define. So even with the organisms like E. coli and bacillus that actually have much bigger training sets or verified operon sets, what I find is sometimes that because obviously they're training this data on these specific existing operons, it would do very well in E. coli and then completely fall apart when they apply that predictor on bacillus, obviously due to things such as overfitting, which things such as implementing user-defined parameters could solve. So I chose Remap and Rockhopper, which both have their advantages, but again, also some limitations. So Remap was chosen to basically lay the foundation of the predictor that COSMO became, because Remap was the first one that actually considered the MTB genome and looked at RNA-seq data. The problem is just that Remap assumed that the expression levels of the genes, or I'll call it CDS for coding sequence, so don't get confused if you hear me say that. It assumed that the expression levels of intergenic regions and the flanking CDSs are the same. And so it actually provides, so for those who don't know, that's the gene or the CDS, and that will be the intergenic region between those two genes. It provided one user-defined cutoff then that would apply to both, which is still better than Rockhopper because Rockhopper gives you absolutely no user-defined cutoffs, and it also uses the intergenic region. But we chose Rockhopper because it outperformed an Operon predictor called DOR 2.0, which actually was performed better than, it outperformed 14 other Operon predictors that they tested it against. So we figured if we can get better than Rockhopper, then obviously, yeah. So one of the problems, another problem of Rockhopper is that it also only predicts operons that are differentially expressed. So you basically cannot really see which ones are active only under untreated conditions versus treated conditions. So some of the questions we asked for our algorithm design was, should there be a minimum CDS coverage? So a minimum average number of reads that would indicate so that we can say this CDS or this gene is expressed. Then is there a correlation between two genes or two CDSs in an operon? Do they stay below a certain fold difference? And then should the intergenic region, so this region again between the genes, should it be an independent parameter? Should it even be a parameter? And if yes, then should it also have a minimum coverage that we would consider to make us consider expressed? And then what would then also be the fold difference or the correlation between an intergenic region in a verified operon and its two flanking CDSs? Do they stay below a certain fold difference? And then lastly, we obviously wanted to confirm, and again, I included this, even though Remap showed it, because all the other predictors are saying this is the most important feature. Intergenic distance is the most important predictor for accurate operons. So I couldn't just exclude it on the basis of one study, sorry, Remap. So I just wanted to test that again. So we took at this stage a few strains from the Beijing lineage or the L2 lineage, which had 3 biological and technical replicates grown under control conditions and then RIF stress. And then at this stage, we had 49 verified operons, which we got from literature. And then we created a simulated set, which we took from random parts of the genome, and it matched the operons in terms of its length and strand orientation. But again, this is not fail-safe because those could be undiscovered operons, but we needed something to work with. We obviously then converted our RNA-seq files or our FASTQ files into RNA-seq files, which we then converted into Wiggle files so that we could pull out those averages. And then we compared these genomic regions. So your UTRs, sorry, the UTR is green, the UTR, which was 300 bases upstream of the operon, to the intergenic region and the CDSs. And when we look at the fake ones, you can kind of see this was more or less commonly what we saw. Either there was basal level transcription or there were absolutely no real correlation between CDSs and intergenic regions. Whereas what we constantly saw in real operons was that the CDSs tended and the intergenic regions tended to kind of correlate with each other. But we couldn't really establish a cutoff for considering the CDS or the intergenic region expressed because, and this is not even one of the lowest ones, but sometimes the CDSs and the intergenic region coverages were quite low. But obviously we did include that as a parameter. I did some multiple regression analysis and it showed that these features were having a significant impact on the dependent variable. So yeah, so the CDS became a parameter that the user can define and the intergenic region, but I'll talk a bit about that further on. We then checked the fold differences between genes of verified operons, and definitely for the real ones, there was a statistically significant fold difference. They tended to remain with just one outlier, basically. They tended to remain within this 5 to 7 times fold difference. Then we needed to see whether intergenic region should even be a parameter. And for that, we tested it against another non-coding region, which is the UTRs. And we saw that for the real verified operons, the intergenic regions were definitely much more upregulated than the UTRs, which was not the case for fake ones. And also, which I'm not showing here, is that we also saw that the intergenic regions were usually much more upregulated or downregulated than, so again, this intergenic region were much more up or downregulated than the 2 flanking genes. So it needed to not just be an independent parameter, but independent from the CDS's coverage. So that became also a parameter on its own. And then, now there's some methods behind this, but I'm time constrained, so I'm just gonna mention that we then obviously checked whether there was a correlation between, or what the correlation should be between the intergenic region, which is, we only take in the middle section of the intergenic region, compared to the 2 flanking ones. And again, they were definitely below, showing that there were some constraints in terms of we should include a maximum fold difference. Finally, we checked intergenic region distance, and although we saw that the lengths of the intergenic regions were generally around the 0 mark, so in other words, the CDSs were mostly overlapping in verified operons, but there was still a large number of these operons that actually had intergenic regions that were larger than 20 base pairs. In fact, when I tested it on the 50 base pair length, not even on 20, I saw that we would actually, from the existing operons that we have, we would completely exclude total 33% of total operons if we include this as a parameter. So that was definitely booted. So I can't really see if you can see, because obviously the Zoom thing is in my way, but we have COSMO, which you can find on GitHub. It takes a RNA-seq file, BAM file, and a GTF file that basically gives you the coordinates of the genes and then 4 user-defined cutoffs. I'm going to explain it this slide. So the user would give a cutoff that considers the CD is expressed or not. If that expression level is above what the user gave, the COSMO then says, okay, this is potentially the first operon. But it then goes on to the next one, which is adjacent on the same strand, the next CDS. If it's also above the cutoff level, it then asks, before we conclude that it's part of the same operon, let's first check if it overlaps. If it overlaps, it's automatically part of the same operon. If not, it looks at the intergenic region and it compares again this middle region. If the intergenic region firstly is, as we see here with B, above the cutoff for minimum expression level, it then says, okay, we're still on par with it potentially being part of the operon, but now let's also compare whether there's a fold difference correlation between the expression levels of the intergenic region and the two flanking genes. If it's still the case, then it starts to add it to the operon after it also checks out fold differences between adjacent genes. And it goes on like that, keeps adding genes. But one thing that it also does, which I have not seen in other predictors, is it doesn't just compare to the previous gene, but let's say, for example, you get to operon CDS5, it needs to correlate with all the genes before it in terms of its expression level. So let's say CDS5 correlates in terms of expression level with CDS4 and 3, but it breaks here at CDS2. So in other words, I'm saying it's below the maximum, it's above the maximum fold difference that must exist between these two CDSs. So instead of just saying, let's break the operon there, it says we encountered our problem here at CDS2. So let's go back to where the problem was and see, does CDS2 correlate better with CDS1? Or does it correlate better with CDS3? If it's with 1, then it says, okay, then these two should be part of an operon, and actually this operon should have been split over here, and it creates the second operon from there. If not, if it correlated better with this one, then obviously it creates this independently and says these one should actually be an operon. Okay, so in terms of results, with just that small subset of lineage 1, we were able to definitely predict more total operons. This is a total operon. Normally what you see in other predictors, even the ones that I tested against, they would predict operon gene pairs and not the total operon, because the largest value that the others have gotten so far, even for Bacillus subtilis, was usually 40% of total. So Remap and Rockhopper definitely already did much better in that sense. And we were obviously able to predict more total operons. So PPV here would be the number of operons predicted under those default parameters divided by the total number of operons that exist. We also were able to predict more unique operons compared to Remap and Rockhopper. And then, of course, what Remap was able to do and Rockhopper wasn't, was it was able to distinguish which genes, which operons are present under control and treated conditions. And obviously, we did a better job at it, probably also because we predicted more operons. Then for the second part, we then combined all of our strains from lineage 2 and 4, which included wild type and rifampicin resistant strains. Ended up being about 64 strains, and we wanted to see, are there operons that are always static, and how are operons changing or being modified under different lineages and conditions. So I think one of the first things I want to say is, when we added the lineage 4 strains, we were happy to see that we were also able to bring our prediction rate up from 60% to 70%. So that was awesome. Just wanted to get that out of the way. But why are we interested in static operons? Well, the logic is that if some operons are always the same length, and by static here, I mean all of them predicted it exactly the same, regardless of the lineage, regardless of the experimental conditions. So all 64 strains predicted it, for example, as false positive. Then potentially they could be used for drug targets. So in this case, firstly, if they're always the same as in literature, then they are most probably sort of what we have with housekeeping genes. They might be housekeeping operons that are under some form of pressure to not change. We would also then potentially be able to predict motifs so that we can better optimize our operons. If they're always longer across all the strains, then potentially they could be undiscovered operon genes, and always shorter could mean that under rift stress, they are actually redundant genes. Similarly, if they are always changing, longer or shorter, and certain genes of an operon are only active under certain conditions, then we could, for example, see how MTB is using those genes, especially if ones are added to circumvent the stress, or even assign functions to hypothetical proteins. If they are changing in a lineage-specific way, yeah, that's also useful. We can see how they are changing their virulence or pathogenicity in regard to the lineage. And then finally, we looked at dysregulation. So average operon expression, is it changing in a lineage or condition-specific way? Because perhaps the operon doesn't want to change its length, but it's targeting the individual genes to upregulate the operon. Why is it doing that? So just to say again, to determine the calls, obviously if there was a unanimous, let's say for example, we're looking at false positive, they all calling it false positive, then we assigned it that call of false positive. If two of the biological replicates had, for example, one technical replicate, up to two biological replicates that had a different call, we would still assign it that call. But when two technical replicates for biological replicates or more started having a different call, then that would be inconclusive. We obviously couldn't have a true negative set because we don't really know which genes are never part of operons. There's no way to really test that. What was surprising was how much, well, it was surprising until I found one article in literature, let's just leave it there, but it was surprising how much it resisted being reorganized. I mean, you can see here this 90%. So here, I mean, we compared the operons that were predicted under control, so untreated conditions, and then we looked at how they changed under rift stress, but within each lineage. And then you can see here 90% of the operons actually didn't change under rift stress. And even the ones that were the most readily modulated, only 30% of them actually were different under rift stress. So there was quite a bit of resistance to change. Then if we go back to what I previously stated, we looked at true positives across the 64 strains. They were always called exactly the same as in literature. Why? When we did analysis on them, they were all operons that were involved in virulence. So that was obviously not surprising. Things such as reactivating MTB from its dormant state, intracellular multiplication, obviously immune response, and drug exporter proteins, not surprising. For those that were always longer than in literature, there was always some reasonable hypotheses for why this was the case. I'm not presenting all of the results, obviously, but I'm just going to show one particular one. This 3-gene operon was always, according to literature, it's a 3-gene operon. We were seeing, COSMO kept on saying this is actually a 10-gene operon. And when we did a string analysis on it, we saw that all except for one, which was still an uncharacterized protein, were actually involved in the same biological process. So again, using these type of predictions, we might be able to see, maybe even assign functions to uncharacterized proteins, and add and see which other genes are actually actively being activated under different conditions. For the false negative ones, this was also the case. Usually the intergenic region was way out of correlation with the CDSs, or there were no reads. One particular study showed, for example, so we always predicted this gene as independent from the other genes. And when we looked at other studies, there were 3 independent studies that actually showed under isoniazid thiolactomycin, and triclosan, this gene also was always highly dysregulated compared to the others. No study as of yet has done it for RIF, so we technically kind of might be the first one. But it was again nice to see that, yeah, the Operon predictor is giving us some valuable information. We then looked at differential expression. How are the operons differentially expressed under RIF stress compared to when they're not under RIF stress? At this stage, we had 50 operons, massive increase. We looked at obviously the log2 FC for absolute 1. And again, all the biological samples had to receive the same consensus for it to be considered up or down regulated. Again, here we were surprised that there was not that much dysregulation. But what we did see, for example, these 3 individual families all called just one operon as upregulated. This is not the real name of the operon. I obviously haven't published yet, so I'm trying to keep it a little bit incognito. And then this one had 2 operons, only 2 operons that were upregulated. And this one was kind of an outlier. It had 25 of the 50 operons dysregulated, with 1 downregulated as well. But one thing that is very interesting, if you notice here, there's 1 operon that was consistently upregulated and shared amongst them. So obviously, I wanted to know what it was doing, and I found, not surprising, that it was the efflux pump. So these patients were actually under treatment at the time that we took their samples, and as we were giving them drugs, those strains were pumping it back out, undoing all of our hard work. But it was nice again to see that it made what the Operon predictor was predicting was making sense. In some cases, it was upregulated as much as 16 times. This specific pump has also been linked to other anti-tubercular drugs, and also for some of the MTB strains that were resistant to rifampicin but showed no mutations in that RPOB drug resistance gene, they were usually upregulating this efflux pump. So that was nice to see again. Finally, we then, just for interest's sake, I wanted to see if I take some strains from literature that were exposed to hypoxia, am I seeing some other operons being predicted? And we saw one particular operon that was always absent under RIP stress, was never predicted under RIP stress, but only predicted for these hypoxia strains. So me again wanted to see what this operon was doing, and I was very happy to find that it was involved in cellular response to self-estimation, and particularly oxidative stress. For those from a biochemistry background, you will know that hypoxia is kind of linked to oxidative stress response. So I was very happy to find that. Again, yeah, very nice to see the Operon predictor actually showing us some meaningful results if we do some functional annotation with it. So I just want to conclude that we showed that we had improved on existing algorithms, even those that that are highly ranked. We also showed that some appear to be static, which could be good potential drug targets if you look at them in terms of housekeeping operons. Others were dynamic in their length, and a few of them even in differential expression. And these could also be useful for not just mapping operons, but also for elucidating some of the functions of those genes and basically seeing what MTV is doing to circumvent a particular stress present at that time. For future work, we want COSMO to be able to accept multiple files, and then to also be able to do differential expression inside of the operon, which means that we will need to optimize it then for normalization. We also want to test it under some additional stresses, and then figure out how we're going to choose which operons to validate in the lab. And then finally, test the algorithm on different organisms. So one of the students for Alan is currently testing it on E. coli. So I'm very excited to see those results. I would like to, this means apparently, maybe Peter, you can tell me if it's true. Thank you in binary. I'm going to thank all of these people, especially Ellen and coffee and internet and money. And yeah, thank you guys. It was long. Thank you for your attention.
I'm trying to simulate wild applause here. I mean, that was really amazing, Tracey. Seriously, that was. I mean, I'm quite excited at your results.
Thank you.
Okay. Mbandi has his hand up for a question immediately.
Okay, Mbandi. Hey, Mbandi.
Hi, everyone. Hi, Tracey. That was a great presentation. And I think that probably the time wasn't enough for you to maybe provide us with some in-depth of the entire process. But this subject is particularly of interest to me because before I left Sanbi, I was kind of interested in looking at intergenic regions for non-coding RNA, which at the time, I don't know what it is now, were of very great interest in MTB. So the first question I have is that, so in terms of Operon, are you referring to only the constitution of genes that are coding, or that also includes genes that may potentially be non-coding? The reason why I'm asking this is because you may potentially have regions in a particular segment of your DNA that is part of the intergenic region, and it may be influencing another gene downstream, and that might in a way reflect in your RNA-seq as if they are part of an operon. So I'm just wondering, how do you address scenarios where an intergenic region where which may be a source for a non-coding RNA might be regulating a gene downstream, and that appears as if that gene has a coordinated expression with other genes from the same region where the non-coding RNA emerge? I don't know if that's
well, are you talking in terms of regulants now? Because obviously, yes, we are defining operons as genes that are adjacent to each other. We are looking at the intergenic region as well, but only, again, it's limited to, because we are not defining the regions, for example, ourselves. We are taking it from a GTF file. So it's largely dependent upon whether that GTF file is basically giving us those coordinates for that RNA, what is it, small non-coding RNA. But the other thing is, if you're talking about the gene that is, are you saying the gene or the intergenic region in this case is far from the genes that we are concluding or that are part of a putative operon, or are they directly next to it? I think I first need to ask.
So take, for example, you have gene A and B, and there is an intergenic region between the two of them. And that intergenic region is a source for a non-coding RNA regulating gene C, which is distal to gene A or B, such that in the RNA-seq data, based on your criteria, the fold difference between A, B, and C are similar, but C is not part of the operon. It only looks similar because there is an intergenic region between A and B that's regulating C. So how do you address this kind of scenario? Would that just appear as a false positive operon in that case?
Wait, so let's say A and C is correlated in terms of its expression with B, but you're saying the intergenic region in between. So remember, we are also looking at the intergenic region expression. So normally what we would see, because I've had a scenario like that where we had a small non-coding RNA in the middle of an operon. That intergenic region expression would be sky high, right? There would be like this huge, and yes, in this case, unfortunately, it possibly could break the operon. if the intergenic region distance or the intergenic region coverage is way above the average cutoff of that the user has given. Is that what you're asking? It's smack in the middle of a putative operon. And yeah, unfortunately, yes, it might break it, although in the cases that we've seen, it didn't. But I've seen instances where the intergenic region or the non-coding RNA would be really, really, the expression level would be really high, but I haven't really seen it inside of an operon. But that's a good question. I don't know yet. I think, so the nice thing about the GTF file, and I think I've scratched the surface of thinking about it, because the non-coding, the GTF file gives a different genomic name or description for the non-coding RNA. So, you know, with the normal, you will have the RV something, but it will have like the E something, that long E number. So I was thinking that we might need to consider that these non-coding, to treat their expression levels differently just based on the name that is being assigned. The problem that we have at the moment is that this Operon predictor is not independently of the GTF file assigning transcription start sites on its own and genomic regions on its own based on just the expression levels that are there. But that might be a good place to start to make sure that we include those intergenic regions and not actually just look at the GTF file. And by including, I mean, the minute that we see that there's this high expression level, that we actually take a further look and see whether we need to do some additional tests because it's a potential non-coding RNA.
Thanks. I could ask another question if there's no one to ask one. So Tracey, can you please just explain how you compute the fold difference? I'm just trying to understand how that relates to similar expression between adjacent genes.
We pulled it out from the Wiggle files, and then we use PySam. So it's basically we are adding up so we're using the same method that Remap basically used. I wonder if can I just escape here quickly? If I can just actually see if I can show in my thesis, or maybe that will make it a bit too complicated. But we basically we take the average of the particular genomic region as defined by the GTF file, and we calculate the average number of reads, and then for example, let's say the average is 10 times, and that's obviously normalized by the gene length. If we then go and look at the fold difference, for example, between that gene and its preceding intergenic region, we would then take that intergenic region as defined by the middle section of the intergenic region. And no, I didn't have time to explain that. And we would also calculate the average number of reads, for example, the read depth for that intergenic region. If the user then said that the maximum fold difference that must exist between these two averages must be 5 and below, then if it's obviously above that, then we would exclude it as part of the operand. I don't know if that answers you, but there is a mathematical formula that is used to calculate the
Great,
reads.
thanks, Chris. Yeah, no, that explains it, because I just wanted to make sure that you know, you take into consideration other aspects of each gene that might make it appear different in terms of coverage, length, and all those components.
Oh yeah, the normalization. Yeah.
Thanks.
Okay.
Okay, are there any more questions for Tracey? I had some very basic questions, but I see we only have a few minutes left. Maybe I can just ask one and then see if anybody else has any questions. I see in your example there where you showed like a very low kind of coverage, Tracey, the RV0097 and RV0102 plus. Could this just be a case of an operon which is actually just expressed at a very low level?
Yes, so that's why we were happy that we didn't just use, so we allow the Operon predictor to or the user essentially, to be able to actually give expression levels as low as 1 or 2, so that we actually press for the fold difference between genes to actually rate a bit higher in terms of then predicting as an operon. So then in that case, we are able to predict very or downregulated operons or operons expressed at basal levels, but just because of the fact that the expression levels of those genes in the operon are correlated and the intergenic regions, we can then still say, oh, these are potentially part of an operon, is just underexpressed, for example.
Okay. Olaton, I see you have a hand up.
Yeah, thank you. So very quick question. I know you reported data of the true positives, true negatives, false positives, false negatives, and such. But then I'm just curious if you actually did calculate from all those variables, the accuracy and other things like, you know, maybe F1 score, sensitivity or specificity of your model in comparison to the other two tools that you had mentioned when you were doing your presentation.
No, so I didn't agree with the methods that the other two predictors used. So there is no true negative set so that we can calculate F1 scores and sensitivity and specificity and all those. And I didn't like the method that they used to create a true negative set, because it doesn't, I don't know, it just doesn't make any sense. Your true negative set in their case will always be true negative. There's no way that the number is going to fluctuate. So I didn't have a true negative set. And then from literature, I saw that in a case when you don't have a true negative set, the positive predictive value is the only next best thing that you can actually use. So no, I was not able to calculate it. I'm hoping that if somehow, because I've got higher, that I can obviously predict more operons, that those numbers will also be better, but obviously I can't account for false positives.
Correct. Yeah, so yeah. My concern with your, you know, statement or opinion is that you need a way to determine that your new predictions are in fact correct. So there's need for some, you know, validation or something. Otherwise, anybody can come up with new predictions, and then how do we determine what's true?
But I've spoken to so many statisticians, and nobody's able to solve this problem for me, because the true negative set firstly is very small, and it's almost impossible. It's impossible to create a proper true negative set, because there's no way to say these two genes are never part of an operon. Conditions may change, and then all of a sudden those genes are part of the operon. So so to create a true negative set for this is just, there's no clear-cut way to do that.
Okay.
So hopefully someone will solve that problem by, I've seen some people employ some strategies. One kind of seemed like it's okay, but I'm just not happy with how they've decided, because they would even they would calculate true negatives, especially from what I've seen with, for example, my comparators, for genes that are on two opposite strands, and I don't see how that's ever going to, I mean, the very nature of an operon is that it's supposed to be adjacent genes on the same strand. So unless they redefine what an operon is, I don't really agree with the existing methods of creating a true negative set.
Okay, sounds good. Yeah, thanks.
Okay. Yeah, I'm hoping, I spoke to that guy in Ghana. I spoke to the other guy, Jay, that was at Sandy as well. I spoke to a few of them, and they said that they agree with me that that way of creating a true negative set just didn't work.
Well,
and besides,
thanks, Tracey.
They were testing. Oh, thanks. One more thing, they were testing their algorithm based on operon pairs and not on total operons. So they were basically saying, you know, if there are 3 genes in an operon, then they would gene 1 and 2, 2 and 3, 3 and 4. And so it was a bit easier for them to just look at the boundaries of the operon and take a gene that was outside of that boundary as a true negative, whereas I'm predicting total operons. So I'm hoping to solve that as well, Leighton. I really want a ROC curve. But at this stage, yeah.
Sounds good. Thanks.
Okay, you're welcome. Peter, I don't think we
Thank you
have time
so
for
much.
breakout rooms.
We do not have time for breakout rooms. In fact, before this meeting, we had such a fascinating discussion about like what we could talk about. And I'll just leave you with some of the questions that we came up with. We were talking about, we always want kind of collaborations. As Leighton said, we need to validate our methods with other things. And, there are so many like really troublesome diseases that we face in the world. So we were thinking about what you would involve in your dream team, okay? Which I then thought about in two ways as well. What is it that you think that other people need to be learning for bioinformatics, or who needs to be engaging more with bioinformatics? And then vice versa, what do we think that bioinformatics needs to learn from? Who must we be learning from? So, obviously there's a lot of the biology of the organism involved in Tracey's presentation. I think that's something to learn a lot from, people who really know the organism deeply. I will just leave you with those questions. We do not yet have a topic for our next event. I've pasted the Slack joining link. If you're not joined on the Slack and you want to join it, in the chat. And now finally, I'm pasting also, if you are not on the mailing list, please join the mailing list. We are soliciting topics for our next meeting, which, let's see, we are in, we are in September now, so we try and do things in approximately quarterly way. So it'll be either near the end of this year or very early next year. Thank you to everybody for attending. Does anybody have any last minute announcements or questions? I'm just going to paste one thing. I am actually leaving this meeting to go join the micro galaxy meeting, which is started at the same time as our one. This is for people doing microbial bioinformatics in a galaxy. Let me just paste the link in case anybody else wants to join. Sorry that pasting on Zoom is not as simple as all that. So the meeting minutes are here. And the Zoom link is coming now. If you're interested in extending the Galaxy ecosystem or working with other people using Galaxy for microbial research, not necessarily pathogen, not every microbe they study is a pathogen. Okay, and that brings us to the end of our time. And thank you once again for attending. We hope to see you next time, and we hope that everybody's signed up to our various discussion forums and actively uses them.
Thank you, everyone. Bye-bye.
Thank you. Bye.
Bye.